lesson for today is all about the parts and the function of the cell. First, we must define what is cell, what are their functions, and what are their parts.
What is cell? Cell is the basic structural, functional, and biological unit of all known living organisms. We have the two types of cell. The first type of cell is the eukaryotic cell. It is the cell that contain a nucleus and organelles and are enclosed by a plasma membrane. Multicellular organisms that have eukaryotic cells include protozoa, fungi, plants, and animals. Eukaryotic cells contains a variety of structures called organelles, which perform various functions within the cell. Eukaryotic cell include plasma membrane, nucleolus, nuclear envelope, nucleus nuclear pore, rough endoplasmic reticulum, smooth endoplasmic reticulum, cilia, Golgi complex, peroxisome, lysosome, microtubule, mitochondrion, ribosome, microfilament, centrosome, flagellum, and cytoplasm. The parts of eukaryotic cell and their definition. The first, the nucleus. A membrane-bound structure that contains the cell's hereditary information and controls the cell's growth and reproduction. Endoplasmic reticulum, the rough and the smooth. Rough endoplasmic reticulum is primarily concerned with the synthesis, folding, and modification of proteins, especially those that need to be delivered to different organelles within the cell or secreted from the cell that we call quality control. Smooth endoplasmic reticulum is largely associated with lipid, we called fat, manufacture and metabolism, and steroid production, hormone production. It also has a detoxification function. Endoplasmic reticulum include ribosomes, nuclear envelope, nucleus, rough endoplasmic reticulum, and smooth endoplasmic reticulum. We have the Golgi apparatus. It receives biochemicals in a bulk flow from the rough endoplasmic reticulum that we call RER. It is the only organelle in the cell that receives, sorts, modifies, concentrates, packs, and dispatches biochemicals for use inside and outside the cell. Golgi apparatus include transport vesicle, secretory vesicle, cell membrane, and secreted proteins. Plasma or cell membrane is the boundary between the cell and its environment. It regulates what enters and exits the cell. Outside of cell, we call cell membrane proteins, carbohydrates chains. Inside of cell, the cytoplasm, protein channel, lipid bilayer. Microtubules are involved in mitosis, cell motility, intracellular transport, maintenance of cell shape, and keep organelles in place. Centrosome is an organelle that is the main place where cell microtubules are organized. Organizing changes to the shape of the cell membrane that allows the membrane to pinch in two during cell division. Centrosome include centrioles and microtubules. Ribosome serves as the site of biological protein synthesis. They link together specific amino acids to form polypeptides and they export this to the cytoplasm. Large subunit is small subunit that we call the ribosome. Lysosomes are organelles that contain digestive enzymes. They digest exist or worn out organelles, food particles, and engulf viruses or bacteria. They are like the stomach of the cell, which are involved in digestion and waste removal. Lysosome include bacterium, receptors, phagocytosis, phagosome, lysosome, phagolysosomes, soluble debris, and exocytosis.
Cytoplasm is a thick solution that fills each cell and is enclosed by the cell membrane, mainly composed of water, salts, and proteins. A framework of protein scaffolds called the cytoskeleton provides the cytoplasm and the cell with their structure, transport dissolved nutrients around the cell, and move waste material out of the cell. Mitochondria referred to as the powerhouses of the cell. They help turn the energy we take from food into energy that the cell can use. Calcium storing. Mitochondria structural features include inner membrane, outer membrane, cristae, matrix. Cilia are eyelashes-like structures, microtubule-based organelles that project from the surface of cells and serves both motil and sensory functions. Flagellum is a whip-like structure that acts primarily as an organelle of locomotion in the cells of many living organisms. Microfilaments. They have roles in cell movement, muscle contraction, and cell division. To make a cell move, one end of the action filament stretches out while the other end pulls itself in tighter. The second types of cell, the prokaryotic cell. It is the single-cell organisms. They are simpler and smaller than eukaryotic cells and lack membrane-bound organelles such as a nucleus. Example, the bacteria and archaea. Prokaryotic cell include flagellum, capsule, cell wall, cell membrane, ribosome, chromosomal DNA in the nucleoid, and pili. Capsule. This additional outer covering protects the cell when it is engulfed by other organisms, assists in retaining moisture, and helps the cell adhere to surfaces and nutrients. Source of nutrition when nutrient supply is low in cell. Cell wall is an outer covering that protects the bacterial cell and gives it shape. It includes plasma membrane, cell wall, and capsule. Pili, hair-like structures on the surface of the cell that attach to the other bacterial cells. The function of the cell. First, the nucleus. Cell organelle that houses DNA and directs synthesis of ribosome and proteins. Next, endoplasmic reticulum. Modifies proteins and synthesis lipids. Rough endoplasmic reticulum. Transport of chemical between and within cells. The organelle is associated with ribosomes. Smooth endoplasmic reticulum, its main function is the synthesis of lipids, steroids, and carbohydrates, as well as the metabolism of exogenous substances such as drugs or toxins. Golgi apparatus modifies, sorts, tags, packages, and distributes lipids and proteins. Plasma membrane separates cell from external environment, controls passage of organic molecules, ions, water, oxygen, and waste into and out of the cell. Microtubules are the movers and shakers of the cell interior. Microtubules are thread-like intracellular structures that are responsible for various movements in all eukaryotic cells. Centrosome, an specified role in cell division in animal cells. Ribosome, it is the protein synthesis. Lysosome, digestion of micromolecules, recycling of worn-out organelles. Cytoplasm, provides structure to cell, site of many metabolic reactions, medium in which organelles are found. Mitochondria. The main function of the mitochondria is to provide energy for cellular activity by the process of aerobic respiration. Cilia. Cellular locomotion movement of particles along extracellular surface of plasma membrane and filtration. Flagellum. The primary function of flagellum is that of locomotion, but it also often functions as a sensory organelle, being positive to chemicals and temperature outside the cell. Microfilaments. It is 3D network inside cell membrane. In muscle cell, actin filaments interact with myosin filaments to create muscle contraction. Capsule. The capsule is considered a virulence factor because it enhances the ability of bacteria to cause disease. It prevents phagocytosis. Cell wall. Protection, structural support, and maintenance of all shape. Pile. It is to attach the cells to surfaces. The structure of the cell. First, the nucleus. The main structure making up the nucleus are the nuclear envelope, a double membrane that encloses the entire organelle and isolates its contents from the cellular cytoplasm, and the nuclear matrix, which includes the nuclear lamina, a network within the nucleus that adds mechanical support, much like the cytoskeleton support the cell as a whole. Second, the endoplasmic reticulum. The general structure of the endoplasmic reticulum is a network of membranes called cristae. These sac-like structures are held together by the cytoskeleton. Third, the rough endoplasmic reticulum. This is an extensive organelle composed of greatly convoluted but flattish-celled sacs which are contiguous with the nuclear membrane. It is called rough endoplasmic reticulum because it is studied on its outer surface, the surface in contact with the cytosol with ribosome. Fourth, the smooth endoplasmic reticulum. The smooth endoplasmic reticulum, its main comprises tube-like structure located near the cell periphery. These tubules or tubes sometimes branch, forming a network that is reticular in appearance. Fifth, the Golgi apparatus. The Golgi apparatus, also called the Golgi complex or Golgi body, it is membrane-bound organelle found in eukaryotic cells, cells with clearly defined nuclei, that is made up of a series of flattened. Sixth, the plasma membrane. The plasma membrane is composed of a bilayer or phospholipids with their hydrophobic fatty acid veils in contact with each other. Seventh, the microtubules. Microtubules are essentially thick protein tubes. Microtubules are bulky, strong spirals of thousands of subunits. Those subunits are made up of protein called tubulin. Eighth, the centrosome. The centrosome is made up of two perpendicular centrioles, a daughter centriole and a mother centriole, linked together by interconnecting fibres. 9. The ribosome. It consists of two major components, the small and large ribosomal subunit. Each subunit consists of one or more ribosomal RNA that we called rRNA molecules and many ribosomal proteins that we called RPS or R proteins. 10. The lysosome. They have a simple structure. They are spheres made up of lipid bilayer that encloses fluid that contains a variety of hydrolytic enzymes. 11. The cytoplasm. All organelles in eukaryotic cells, such as nucleus, endoplasmic reticulum, and mitochondria, are located in cytoplasm. 12. The mitochondria. The membranes create two components. The intermembrane space, as implied, is the region between the inner and outer membranes. 13. The cilia. Cilia structure are microscopic hair-like structure that project from the surface of many eukaryotic cells. 14. The flagellum. Bacterial flagella are helically shaped structures containing the protein flagella. 15. The microfilaments. Structure of microfilaments are primarily composed of polymers of actin by and interact with numerous other proteins. 16. The capsule. Capsule structure can be split into two main groups. A proper capsule or capsular polysaccharide, or we call CPS, in which polysaccharide material is attached to the cell, or an exopolysaccharide that we call EPS. 17. The cell wall. Although primary or secondary wall layers differ in detailed chemical compositions and structural organization, their basic architecture is the same, consisting of cellulose fibers of great tensile strength embedded in water, saturated matrix of polysaccharides, and structural glycoproteins. Last, the pile are short hair-like structures on the cell surface of prokaryotic cells. They can have a role in movement but are more often involved in adherence of surfaces.